Okay, good morning, good afternoon, good evening. This is Dr. Kaz here, and this is the remaining material for our Chapter 4 um, uh, class material on the human cell. So we were talking about the different processes, and we left off talking about our passive processes. We completed those. And now we are going to move on to the active processes. So a couple things just to review about active processes. All right. One is most important that they do require energy. So one of the things that we learned about passive processes that they do not require energy, well, active processes do. And one of the things that we learned about passive processes is that we're moving a solute or material from an area of high concentration to low concentration. So we're going down our concentration gradient. In this case, all right, when we're talking about active transport, we have to move a solid or material up its concentration gradient, or what we would say against its concentration gradient. That means we're going to move some material from an area of low concentration to high concentration. And usually, all right, when we're talking about these two areas, we're going to be talking about inside the cell and then outside the cell, which is going to be the interstitial fluid here. All right, so we're going to talk about some of these processes here, right? So first of all, when we discuss that it requires energy, of course, cellular energy is going to be ATP. So what we'll see in this case is ATP will be broken down into ADP. And so that one phosphate molecule that came off of our ATP molecule is going to be added on to a protein. And that process is called phosphorylation. All right, excuse me, phosphorylation. When you hear that term, and we're going to be using that throughout some of the chapters in this semester and then some into 11, just think when you see that term that we're taking phosphate and then adding it onto something. In this case, we're going to add it onto our transport protein. Okay? It could be a pump, could be a channel. And what that does is that's going to change the shape of that specific protein. And in doing so, right, we're going to move whatever it is that we are moving from inside the cell to outside the cell or vice versa. All right, so let's take a peek at that. We're looking here at the ion pumps that are located in our erythrocytes, our red blood cells. So we have a calcium ion pump that are located in the plasma membrane of the erythrocytes. And their job is to move calcium from inside the cell to outside the cell. And in doing so, it helps to prevent the erythrocytes plasma membrane or the overall cell itself to become rigid, because what will happen is calcium has this ability to accumulate in areas, and when it accumulates in areas, whether it's in tissue, inside cells, it starts to kind of harden like a quick drying cement, and it will decrease the cell's rigidity. And our red blood cells have to squeeze in and out of some really tight spaces. So we can't compromise the plasma membrane's flexibility. And so one of the ways that we can make sure that doesn't happen is having these ion pumps constantly moving calcium from the internal portion of the cell to the external portion of the cell. Another one of our transport proteins here are the sodium potassium ion pump. Now we'll get into a lot of detail about this when we're in chapter 12 when we're talking about the nervous system, but also we'll talk about it a little bit in chapter 10 when we talk about muscles. But again, when we're dealing with this type of transport protein, it is a pump, it is an active process, so we will use energy. Okay, in this case, what we'll see is one ATP molecule is going to move, right, 2 potassium ions into the cell. Where at the same time, okay, we're going to move 3 sodium ions out of the cell. And that's all, we get all of that with one ATP molecule. So again, with both of those, we are going to be moving those ions against their concentration gradient. And so the plasma membrane allows us to maintain this gradient because, as you recall, it is a selectively permeable barrier. And so it's going to let certain things in and certain things out. So this is how we're going to be able to move a lot of our cells, our neurons, for example, how our neurons are going to make sure that most of the sodium, all right, that's on hand is going to be outside the cell, and that most of the potassium that's on hand will be inside the cell. Show you a quick picture here to kind of illustrate my point. Right here you can see, here's our plasma membrane transport pump here. And so what happens is you have some sodium on the inside of the cell that the cell wants to get rid of. So the sodium enters into the pump, ATP binds on, right? Then we'll see what happens is that ATP gets converted to ADP. We take that phosphate ion, attach it to the pump, right? That causes the pump to change shape, causes the sodium to leave from the inside of the cell. Then what we'll see is the potassium that's sitting outside the cell in the interstitial fluid, that will gain entry into the pump. And then as soon as that phosphate ion pops off of the sodium potassium ion pump, that pump is going to go back to its original shape. And in doing so, it allows the potassium to enter into the cell. And then the cycle repeats itself. And that's all on one ATP molecule. That's what we're going to see here. And so this is constantly happening. And this process, and we'll talk more about it when we get to the electrochemical gradient of a neuron. But by design, we want to continue to move sodium out of the cell and move potassium into the cell. Okay, so of our active transports, we have another type of active transport we call secondary active transport. Same processes, okay? It requires energy, and we're going to be moving substances against their concentration gradient, right? So, or in other words, we're going to move substances up the concentration gradient. And the nice thing about this type of active transport is that we're going to be moving two substances instead of one. And another interesting fact is when we move one substance, okay, during this active transport, it's going to generate kinetic energy. And that's energy that we can use to move the second substance. So what happens with this type of active transport is that one of these substances has to move down its concentration gradient, right? And moving down its concentration gradient, the movement of that substance, molecule, whatever, is going to generate kinetic energy. And then that kinetic energy will allow the movement of that second substance up its concentration gradient. So there's two types, symport and antiport. Sym means that both the substances that are going to be moving during this type of active transport are going to move in the same direction. Sym, same, right? Antiport, two substances will be moved, but again, we'll see, okay, with the movement of these substances that one is going to go in the opposite direction of the other. So anti will be opposite of one another. All right, let's talk about the active processes of vesicular transport. Another name for that is bulk transport because we're going to be moving a lot of stuff during this type of transport here. So again, we're going to be involving ATP energy, right? When we have to move these transport substances, right, called vesicles. And we recall, right, at the beginning of this chapter when we were talking about the endoplasmic reticulum and the Golgi apparatus, how the Golgi apparatus will package up the proteins that it's modifying, all right, and helping to contribute to making, and it packages up into a secretory vesicle. Well, again, a vesicle is just going to be, right, a sac, a membrane-bound sac that has the stuff inside of it that we just made. So there's two types of processes, exocytosis and endocytosis. Think exo, exit. We are exiting the cell. So when we're talking about exocytosis, say our cell has made some macromolecules, maybe some lipids, maybe some carbohydrates. Proteins we'll use because proteins, for my example, we use those quite a bit. So in this case, our cells made some proteins, and they're just too big. We can't get them across the plasma membrane. So we're going to package them up, okay, like we talked about with the rough endoplasmic reticulum and the Golgi apparatus. So it's going to package up this material, and it's going to put them into our transport vesicles, which are our transport vehicles that move throughout the inside of the cell. And what will happen is those vesicles will make their way to the plasma membrane and eventually fuse with the plasma membrane. In doing so, that is the energy, all right, required step. That fusion of that vesicle to the plasma membrane requires ATP. And once it fuses, then that vesicle starts to open up, and the contents that were inside will then be released to the external or the exterior of the cell. And we see that, and we'll get into it quite a bit when we get into chapter 12 about the neurotransmitters, right? And those are those chemicals that our neurons store. And when an action potential, which is a nerve signal, moves along the cell, it will eventually trigger this process of exocytosis, right? These neurotransmitters are stored inside the cell in their vesicles. And then when that nerve signal comes down, it will trigger the movement of those vesicles, all right, to go to the plasma membrane and then fuse with it and release the contents, which are the neurotransmitters, through that process of exocytosis. All right, so let's take a peek at that. What does that look like here? All right, so here you can see, here's our secretory vesicle with its manufactured products inside, and it migrates towards the plasma membrane. And then it fuses with the plasma membrane. This is where our ATP is required. And as it fuses, it starts to open up. And you can see we'll start to expel the contents that were inside of that secretory vesicle into the interstitial fluid or the extracellular space there. And that's what, well, I skipped one, sorry. You can see it starts to open up here, and then it expels the contents. So that's exocytosis. So that's getting stuff out of the cell. Well, endocytosis is when we're trying to get stuff that's outside the cell into the cell. Endo, enter. All right, so in this case, we're going to be, again, going after some relatively large substances. And think of that process that we just talked about with exocytosis, but reverse the order of everything. So in this case, our plasma membrane is going to create these pockets or these invaginations. And they'll push in towards the internal part of the cell into the cytoplasm. And the plasma membrane will pinch off and it will form its own vesicle. So a lot of times, cell wants something outside, like it sees some delicious carbohydrates or maybe some lipids. Lipids is a different story, or some carbohydrates or some fluid droplets, it'll pull it in, right? Or sometimes, right, how do we get that plasma, that secretory vesicle, how do we extract this secretory vesicle, all right, from the plasma membrane? We'll utilize endocytosis for that process there. Right? And we're always adding things to our plasma membrane and then taking things away from the plasma membrane. Endocytosis is another example, not another example, but a way that we're able to do that. So let's talk about some of the types of endocytosis. There's three types: phagocytosis, pinocytosis, and then receptor mediated endocytosis. Phagocytosis, we've talked about this before when we were talking about the macrophages. Right? It's essentially when a cell goes around like the Pac-Man from the video game and eats things. So part of the cell is going to extend itself, forming these pseudopods, like these little arms, and it'll reach off from the plasma membrane and circle whatever it is that the cell wants to take in, and then it'll form off a vesicle or a membrane sac, and it'll pull that membrane sac into the inside of the cell. And normally what will happen is that membrane sac that it forms will fuse with the lysosome, and then the lysosome will empty its digestive contents into that sac and then break down whatever it is that's in there. right? That's one of the ways that your thyroid gland, all right, makes thyroxine. All right, so it will, that's how it, that's part of the process when it's trying to get iodine into the cell there. All right, so a couple cells that that will do this. Mainly they're going to be the white blood cells there, especially the macrophages, but that's what they do. That's what they're programmed to do. They're supposed to go around, find things that shouldn't be there, all right, ingest it, and then kill it or digest it. So that's what we see here. Here's our particle that our cell wants. It's extending the pseudopods. Those pseudopods are now surrounding that particle, and then it pulls it in. And then eventually a lysosome will come over and then fuse with that and then pour its digestive enzymes. So it doesn't necessarily have to be a lysosome every time, right? There's other cell organelles that might be a part of that process, whatever it's going to be used for. So that's phagocytosis. Penocytosis, right? We talked about eating. What about drinking? Same type of thing. Okay, we have some droplets that are outside the cell, right? In this case, it won't be one big vesicle. We'll make many smaller versions of that, right? And then what will happen is, all right, the cell will ingest whatever it is that it's going after, all right? This process is relatively non-specific, okay, because a lot of times when we're talking especially about lipid or liquid droplets, right, it's not like you have, right, liquid droplets of urea concentrated in one area and then liquid droplets of let's say carbon in another area. No, they're scattered throughout. So it's pretty much you get what you get, you know, and you don't pitch a fit. You just ingest it, take it in, and then digest everything. And then our last form, all right, here of the vesicular mediated active processes is receptor mediated endocytosis, right? So of course, when you see the term receptor, we're going to utilize receptors on the plasma membrane. And so what happens is, right, the plasma membrane has all these various receptors on the outside of the cell, and some sort of molecule, all right, will bind onto those receptors. And then the cell will then, all right, absorb, all right, that part of the plasma membrane, all right, receptors and all, and everything that's bound onto those receptors, and it will actually create its own secretory vesicle, not secretory vesicle, but its own vesicle. And in this case, it'll put on a special coating. They call it a clathrin coating. And it's just like a tag. It identifies specifically, all right, what that vesicle is. Now we see this quite a bit when we're trying to move, and we'll learn about this more in 211, how cholesterol, right, that's floating around in your blood will enter into a cell. So I'm sure that some of you that when you've gone and gotten your blood work done and they do a lipid profile on you, you heard terms like good cholesterol and bad cholesterol, right? Well, the bad cholesterol, the LDL, which stands for low density lipoprotein, is basically, you know, a protein with lipids attached to it, right? Well, they float around and they attach cholesterol to them. And so then these LDLs will come in contact with the LDL receptors on specialized cells, like in the liver, all right? And then they'll pull the LDL with the attached cholesterol to it into the inside of the cell. Something like this. Okay, so let's pretend, all right, these are our cholesterols and our LDLs that are attached to them. Then we have these special receptors there, and then they attach onto the receptors. The plasma membrane then engulfs, right, those LDLs with the cholesterol, and then creates its vesicle, and then it coats the outside of the vesicle with this protein called clathrin, so we can identify, hey, this has cholesterol and some LDLs inside of it, and we can use it for whatever processes that we need to undergo. Okay, let's just, I want to quickly mention resting membrane potential, since we are talking about the movement of substances across the plasma membrane. We will talk about this quite a bit in detail when we get into chapter 12. Okay, but for right now, I just want to give you a kind of a basic rundown. of what the resting membrane potential is. Our plasma membrane is basically, like I said, is going to separate, okay, contents outside the cell from the contents inside the cell. And in doing so, it is going to establish what we call the electrochemical gradient, meaning certain chemicals will be in higher concentrations outside the cell or inside the cell. And in that happening, it will generate, okay, a difference in an electrical charge. Again, if you don't really quite grasp that, that's not a problem. I'll go into that in more detail. But point being is the plasma membrane will create this, what we call this electrochemical gradient. And when a cell is not doing anything, when a neuron specifically is not doing anything, okay, we call that the resting membrane potential. So like when you're sleeping tonight, right, you're not going to be using your sciatic nerve to move your leg around. So the neurons in the sciatic nerve will be at rest. They won't be doing anything. So the neuron, the plasma membrane of the neuron is going to establish this resting membrane potential. And in doing so, it's basically keeping certain chemicals, molecules, right, in greater concentrations inside the cell or outside the cell. And by that occurring, it will create a difference in a charge, right? And when I say that, a charge, right, the outside of the cell will be positively charged and the inside of the cell will be negatively charged, like a battery. You look at a battery, it has a positive end and a negative end, all right? Well, that's what we're going to see here, right? So this is crucial because without it, our muscles won't work and our nerves certainly won't work. So we need to have, all right, this electrochemical gradient in existence. Because if not, then we're going to have some problems, big time. So when we're talking about, okay, this electrical charge that is going to be found across our plasma membrane, basically outside the cell is going to be positively charged. Inside of the cell is going to be negatively charged. And because of that, okay, that difference, that charge difference, we call that the membrane potential. Okay, and so what is a resting membrane potential? That's when the cell, like I said, is not doing anything. Okay, when our neuron is just hanging out like it will be when you're sleeping tonight. So that leads me to this. You're probably wondering, Dr. Kaz, what the heck does banana floating in the ocean mean? Again, that is something I'll get into more detail when we get into chapter 12. But for right now, I want you to think of a banana floating in the ocean. And I'm not going to answer this question. I'm just going to throw the question out there. What is a banana high in? People will say, oh, eat a banana because it's a good source of this. Okay. So if you know the answer to that, all right, it's here on the screen. Okay. If you know the answer to that, right, then you'll understand what I'm trying to get at. The inside of the cell has more potassium. Bananas are a great source of potassium. So when you're thinking the ocean, you have to think of the water in the ocean and what kind of water it is. It's salt water. And if you recall, all right, salt, all right, the molecular formula for salt is NaCl. Okay, so sodium and chlorine, or chloride, I should say. All right, so the ocean is full of salt water. So outside of that banana, that's going to represent the interstitial fluid. So outside of our cell, we'll see more sodium and more chloride outside of the cell in the interstitial fluid. So banana floating in the ocean. All right. So what we're seeing here, the plasma membrane has created this difference, all right, or this unequal distribution of these substances, both inside the cell and outside the cell. So more potassium in the cytoplasm and more sodium in the interstitial fluid when we compare it to the inside of the cell. And this is all due to these guys, these transport proteins, the sodium potassium ion pumps. They're constantly pumping all the time. And no matter what is going on, these pumps, their job is to, like, you're in a boat bailing out water, all right? Well, these pumps are pushing sodium outside of the cell and moving potassium inside the cell. And because we have unequal distributions of these ions and molecules, it will cause, all right, positive and negative charges to be established across the plasma membrane. And that's what we see here, right? Outside of the cell is more positive than inside the cell. So if I draw my line here, it'll be positive outside the cell and then negative inside. Okay, we'll get into that more detail, but I just wanted to throw that out to you a little bit just to kind of give you a little bit of something to kind of refresh yourself on. And that's what this is showing you here. We'll see this picture again, all right? This machine that we have pictured here, that's our voltmeter, okay? And so we put one lead inside the cell and another one outside the cell. And it's just measuring, all right, the difference in charge across the plasma membrane. And you can see, all right, here throughout, positive outside, negative inside. And if you look, all right, those purple balls, that's sodium. You see a lot more purple balls outside of the cell. And then the blue balls, all right, that's our potassium. And you see a lot more of those inside the cell. All right, so we'll get into all that. And then we have, of course, in this picture, all the different transport proteins. We have leak channels there, you can see. All right, they're the ones that are always open. Okay, so we're going to see the movement of ions going through those leak channels, right? And that's all based on the concentration gradient. And then we have our pumps that are always going to be moving 3 sodium out of the cell and 2 potassium into the cell all the time. All right, we'll get into more detail on that when we get to chapter 12. But let's finish up chapter 4 and talk about the last part of our cell. All right, we talked about the cytoplasm and everything in the cytoplasm. We just finished up the plasma membrane. And now the third structure found in a cell is the nucleus. This is when you're looking at a cell on a microscope, this is the largest structure inside the cell. We also refer to it as the control center. Okay, now pretty much for most of the cells in the body, right, you'll have one nucleus per cell. Right, some cells will have many. We saw that with the skeletal muscle, and we refer to that as multinucleated. Even cardiac cells can have multiple nuclei. Okay, they can have up to 2. And then there's some cells that don't have any. Your red blood cells, a mature red blood cell does not have any nuclei. Right, so they don't live very long. And so again, most of the cells will have 1 nuclei per cell. Right? And of course, when we were talking about in chapter 5 with tissues, that these nuclei will pretty much mimic the shape of the cell. Right? We saw with the squamous cell that the cell is flat, and so is the nucleus. Right? But an important thing, and I save it for last, is that our genetic makeup, our DNA, lives inside the nucleus. So everything that your body, it's like the instruction manual on how to make you, okay, is inside the nucleus there. So let's look at the nucleus here, okay? Similar to our cell, you know, the cell had a plasma membrane that went around, okay, and separated the internal contents from the contents outside of the cell. Well, we have a nuclear envelope, and it's also a phospholipid membrane, and it surrounds the nucleus. And it separates the nucleus from the cytoplasm. Well, on the inside of the nuclear envelope, we also have a fluid in there, and we call that nucleoplasm. But the nuclear envelope has these holes in it. We call those nuclear pores. And so there are these little openings, all right, formed by proteins. And so they allow for things to move in and out of the nucleus. Right, so it can be some of the molecules that are inside, like parts of the ribosomes, are inside of the nucleus there. And we have to move those outside of the nucleus because we want to use them for protein production. Right? But also ions, water, they'll move through, okay, these nuclear pores. All right. So Inside, this is almost like one of those Russian doll things. You know, you open one thing up, you'll see a smaller version of the doll. Then you open that doll up, and you see a smaller version of the doll. Well, inside of our nucleus, we have a smaller structure called the nucleolus. This one doesn't have a membrane around it. Okay, it does not have a nuclear envelope. All right, but what we'll see in the nucleolus here, all right, we've got proteins, RNA, and this is where those ribosomes are made. So the small and large subunits for the ribosomes are made in the nucleolus, and then they will leave the nucleolus and be assembled, okay, put together so they can start to produce, all right, proteins. All right, so we won't see a nucleolus in every single cell. All right, sperm cells, for example, all right, will not have a nucleolus, and in some, all right, we will see that we will have more than one, okay? All right, so here you can see on our electron microscope here, all right, we're looking at the nucleus, which is this structure right here, and then you can see the nucleolus inside. So here are the nuclear pores located in the nucleus, and these nuclear pores will be, you'll see how they'll have, well, it's not really seen very well here, but they will form, all right, connections to the endoplasmic reticulum. So things can move in and out, again, because we have to make certain substances. All right, so one of the last things to talk about in the nucleus is going to be our DNA. And what is the DNA? All right, so DNA, like I said, is our genetic makeup, the blueprints or the instruction manual on us. All right, so first of all, what is DNA? Okay, well, it stands for deoxyribonucleotide. All right, so what is all that? Okay, so if we break it down, all right, our DNA molecule is going to be made up of a sugar molecule, okay, called deoxyribose, a phosphate molecule, and then one of four what we call nitrogenous bases. Okay, so it's either going to be adenine, cytosine, guanine, or thymine. That is a DNA molecule. And so inside of the nucleus, right, we will string these molecules on end and attach them to one another, right, in the form of what we call monomers. And we've seen that before, like with glucose. Okay, glucose, when we store it as glycogen, we just stick one glucose molecule to another one, to another one, to another one. Okay, well, in this case, when we, all right, the monomers, we call those nucleotides, but specifically they're deoxyribonucleotides. Right, so we're just going to string those together. I'll show you a quick picture here. Okay, so here, all right, is part of, this is the helix structure or the Watson-Crick model of what DNA looks like. And it's a spiral, like a ladder, but we just twisted it around. Right, so this is what we're seeing here is part of our DNA. That's just one, okay? Well, one part of it, I should say. So let me go back. I don't want to confuse anybody. All right, so what we do, all right, with those nucleotides, okay, we are going to attach them to one another, and we're going to do so through what we call the phosphodiester bonds. All right, that's how we'll attach the nucleotides to one another. But if you saw on that picture here, you'll see, you notice how we link them to another DNA molecule across the way. Right, and we call that a complementary pairing. So we are going to pair, all right, two strands together. And because of all this, what we've done is we've created this spiral ladder. And so the rungs of the spiral ladder are going to be the bases that we talked about. What are the bases? One of these four right here. These are the nitrogenous bases. All right, it'll be one of those, right, which will be in the pairing there. And then the backbone, or what we call the struts of the ladder, that's the up and down part, is going to be, all right, the sugar molecule, and the phosphate molecule. So what holds, all right, one complementary strand to another? Hydrogen bonds. And this whole configuration, all right, allows us to put all of our genetic information into each and every single one of our cells. And so for humans, we have what we call 46 separate double-stranded DNA molecules. Or you might hear terms like humans have 46 chromosomes, right? So that leads me to the next thing. You know, what is a chromosome? Well, chromosome, or before I define what a chromosome is, let me define what a chromatin is. All right, so the cell is either dividing or not dividing. In a cell that is not dividing, okay? The form that our DNA is going to be in is going to be this small, very fine filament, okay? And so we call that chromatin. But when the cell divides, now what we do is we coil up all of that chromatin, okay? And then we call it a chromosome. So you ever wonder how we're able to fit all of that genetic information? Because it could go on for miles. So what we've done is we take, all right, that genetic, our DNA molecules, all right, that double helix ladder structure, and we just keep twisting it and winding it around these specialized nuclear proteins. Right. When I was learning this, it looked to me more like a hockey puck. So it's, to me, I was thinking all I'm doing is taking a string and wrapping it around a hockey puck, right? Well, these nuclear proteins are called histones. And then what we'll do is we'll attach these histones together and form these structures called nucleosomes. That brings me to this picture here. All right, so here, you can see, hold on. Okay, you can see here, here's our double helix structure with our DNA complementary, with the complementary strands. We've just, you know, it's twisted here. And so what we've done now is we've taken that and we take that DNA and we wrap it around these individual histones here, all right? And then we coil that up into these nucleosomes. And so all we're doing is just basically coiling everything, coil it up, and then we wrap it up into this chromatin in a non-dividing cell. And then when the cell is getting ready to divide, then we're going to wind up with a chromosome here, which we get this clumping here of that genetic material. Again, you'll talk more about this. I believe it's chapter 29 and 211. I know I say that a lot. We'll talk more about this in 211. All right, but a lot of what I do here in this class is to lay the foundation, right? Just to kind of get you started, right? So when we get into our DNA and our genes here, all right, yes, we're going to see that it's a repeating, not a repeating monomer, but made up of many monomers, all right, those nucleotides here, right, that will basically give us instructions on how to do things, how to make stuff, specifically like making certain proteins, right, and certain genetic traits that you might have, like the color of your eyes, the color of your hair, how tall you are, right, how much melanin, all right, your melanosomes are going to be able to produce. All that information, okay, is going to be stored here in your DNA. So when we want to make something, okay, your ribosomes have to know when they need to start, like where do we start to make a protein and how do we stop, right? So that's what we're going to see. In our DNA, you will have certain areas that tell, all right, the ribosomes when to start to make a protein. And then you have certain areas in your DNA that tells ribosomes when to stop making a protein. It's very helpful. Okay, and there's a certain genetic sequence there of our nitrogenous bases that will tell, all right, your ribosomes when to start and to stop. Which brings me to the last part of chapter 4, that's cell division. Okay, like I said before, we're not going to get into the specifics, but I want you to know the difference between the types of cell division. So I like to start off with meiosis, okay? When our sex cells divide, right? That includes sperm and the oocytes, which are the eggs. Right? When those sex cells divide, okay, that's meiosis. And then mitosis is the cell division that occurs in all the other cells, right? And we call all the other cells somatic cells, cells of the body there. So when we see with somatic cell division, and we talked a little bit about it when we were talking about the stratum basale and the epidermis of the skin with the keratinocytes, right? How one cell will divide. You have two daughter cells, and one daughter cell stays in that stratum basale, and the second daughter cell advances into the more superficial, right, closer to the surface there, to the next strata. So that's what we'll see. With somatic cell division, we'll start off with one cell, it divides, and we end up with two cells. Right? And this goes on and on for several reasons, all right? As you're growing and developing, okay, obviously you can see it for any of you that have young children, okay, when they're creating new tissue, all right? Any tissue that's growing, right, this is necessary. If some cells start to get older and die off, we have to replace them. Right? Or if something were to happen, you were running, you fell down, you scraped your knee, all right, you damaged yourself, well, we have to repair that tissue. So we'll see somatic cell division occur there. So question for everybody. What is the function of our nuclear pores? Give you a second to think about that. Function of the nuclear pores are going to be to allow the passage of particles in and out of the nucleus. All right, folks, that is it for this recording. I hope you learned something. I hope you enjoyed it. Have a great day.